This is your benchmark review for biology. You can access this review as well as this video review at www.srhsbio.wikispaces.com. First section that we're going to work on focuses on DNA and protein synthesis. So first question asks us, how are genes related to proteins? So the first thing you need to know is that genes are the instructions for the synthesis of proteins. The proteins are what leads to our individual unique traits. So a gene leads to a protein, and that protein leads to a trait. So to show you how a gene can lead to a protein and how that protein leads to a characteristic, we're going to talk about how a firefly's tail makes light. So in the nucleus of each cell inside the firefly's tail are enzymes which takes DNA and codes for specific genes. So there's a gene inside the cell's DNA that specifically codes for a enzyme which causes the firefly's tail to light up. So here we are inside of our nucleus, and we can see right here that's the gene that we're interested in, and this RNA polymerase is going to go ahead and make a copy of that gene. So this molecule, this mRNA that it's going to make, is very similar to DNA, except that it's single-sided and it has a different sequence of bases. Instead of A, T, G, and C like we find in DNA, we're going to have A, U, G, and C. So that U is replacing the T. So the process where this RNA polymerase copies the section of the gene that we are interested in into an mRNA molecule is called transcribing. So there we are making our mRNA copy of the gene that causes the firefly tail to light up. Now that we have made this mRNA, it's going to move out of the nucleus and enter the cytoplasm. Once in the cytoplasm, this mRNA is going to meet up with a ribosome. This ribosome is the protein-making machinery of the cell. So there's mRNA attaching onto the ribosome. So again, this mRNA is just for one specific gene that codes for one specific protein that leads to the firefly's tail being able to light up. Now the ribosome is going to read the mRNA in sets of 3, so it will read 3 mRNA bases, and it's going to produce a string of amino acids. Now the way this works is a tRNA molecule is actually going to come and carry that specific amino acid to the ribosome. This process is called translation because we are translating the mRNA into a protein. So there you go, those little circles are the amino acids that we've produced. So this is the enzyme that's going to work in the firefly's tail to make it light up. Now in order for this enzyme, for this protein to be able to function, it's got to take on a certain shape. So other proteins within the cell are going to help it take on that specific shape. Now that we have a completed functional protein, that protein participates in a chemical reaction which changes the shape and the form of the molecule into another type of protein, and that protein is the protein that glows. The reaction that occurs with this enzyme gives off light. So that's why fireflies are able to give off light from their tails, because all of these cells are expressing a particular gene which codes for the protein that lets them light up. The next question asks us about what the building blocks of proteins and DNA are. So the building blocks of proteins are going to be amino acids. Several amino acids strung together make up a protein. For DNA, it's made of nitrogen bases. So examples of amino acids would be like methionine, lysine, etc. Anything that comes from that codon wheel, if you string a bunch of those together, you're going to get a protein. DNA, however, is made up of the nitrogen bases A, T, C, and G. Now in a protein, 3 bases from an mRNA molecule will code for one amino acid. So you use that codon wheel to determine which amino acids are being coded for by the mRNA. DNA makes up the genetic code that is used in the process of transcription and translation to make a protein. Many proteins are enzymes, and enzymes are sort of the workhorses inside of cells. They make many chemical processes possible. And DNA is important because it holds your hereditary information. So there's a picture showing a protein. Again, I've got amino acid, amino acid, amino acid, a whole bunch of amino acids strung together to make a protein. And there's a picture of the double helix of DNA. Next question asks us to describe the function of the 3 types of RNA. So the first type of RNA that we're going to talk about is messenger RNA. Messenger RNA is sort of the working copy of DNA, and it's made from one side of the DNA. It carries the code for proteins in its sequence, and it travels from nucleus to ribosome in the cytoplasm. So there's a picture. You can see that this is my mRNA strand. It was made based on 1/2 of the strand of DNA, this template strand. Once I've made this mRNA, it's going to leave the nucleus, that's where the DNA is, leave the nucleus, head out to the cytoplasm where it's going to meet up with the ribosome.
This animation demonstrates how the digital information encoded within DNA is used to direct protein synthesis. This is a DNA double helix containing the digital code which directs the cell in all aspects of operation. And here we see a protein complex called an RNA polymerase traveling down the DNA strand. As it moves down the strand, it carefully unwinds the DNA, preparing it for transcription. Inside the polymerase, we see a single-stranded copy of the original instructions being assembled as individual bases are positioned and added to the growing strand. A stop code marks the end of the protein specification, at which point this copy, known as a messenger RNA transcript, exits the polymerase and heads towards a 2-part chemical manufacturing machine called the ribosome.
So the next type of RNA that we're going to discuss is tRNA, and tRNA is standing for transfer RNA. Each tRNA molecule has a job of carrying a specific amino acid to the ribosome, where the amino acid is added on to the growing polypeptide protein chain. A set of 3 bases in the mRNA matches to each of the tRNA molecules. So each tRNA molecule corresponds to one amino acid, which corresponds to a particular set of 3 bases. When 2 tRNA molecules are next to each other in the ribosome, a peptide bond forms between the amino acids to form the protein chain. So again, here we have tRNA. You see that the sets of 3, the bases here, are different for each of the tRNA molecules, and they each correspond to a different protein. So here's 2 proteins here. The next protein is going to come in on the tRNA. Whenever these 2 tRNA molecules are next to each other in the ribosome, so we got 2 right here, there's going to be a polypeptide bond formed between these 2 amino acids. So that's going to lead to our protein. The last type of RNA that we're going to talk about is rRNA, which is ribosomal RNA. That's what the ribosome is made of. So here we have a ribosome, and inside of the ribosome I have a tRNA, and there's my amino acid chain that's growing and being formed, and there's my mRNA, which is coding for the amino acid chain. This subunit, this whole ribosome, is made up of our RNA. So the next question, we are given a DNA strand, ATTCGGAC. I want to know what the corresponding RNA code is. So whenever we go from DNA to mRNA, we got to remember that A pairs with U, T pairs with A, and G pairs with C. So every time I see a U in DNA, that codes, every time I see an A in DNA, that codes for a U in mRNA. Every time I see a T, that means that the mRNA is going to have an A. Every time I see a C, I should have a G in the mRNA. A G in DNA means a C in mRNA. So basically the only trick you got to remember is that instead of putting T in mRNA, you should change that to a U. So there's a picture showing you RNA and DNA. RNA is single-stranded. Its bases are A, U, G, and C. And DNA is double-stranded, and its bases are A, T, G, and C.
So now let's talk about the structure of DNA and the structure of RNA. This would be RNA on one side, and this would be DNA on the other. The one thing that jumps out is that RNA is a single helix and DNA is a double helix. But if we look at the structure of both of these, which are called nucleic acids, they're mostly made up of just 3 parts. And so both of them have, let me get a color that works, both of them have a sugar. In RNA, that sugar is going to be ribose sugar. In DNA, it's going to be deoxyribose sugar. They then have a phosphate group, and then they have bases on the inside. And so if we look at, this would be RNA right here. On RNA, you have a phosphate attached to a ribose sugar, attached to a phosphate, attached to a ribose sugar, attached to a phosphate, ribose sugar, phosphate, ribose sugar. And so the backbone, which on here would be this kind of greenish part that goes down here, is simply a phosphate attached to a sugar, attached to a phosphate, attached to a sugar. That's relatively boring. It's the same all the way down. Now if it's DNA, it's going to have a deoxyribose attached to a deoxyribose attached to a phosphate, deoxyribose, and it's going to be the same side on both sides of that backbone. What goes off the inside attached to the sugar is going to be the base, or the nitrogenous base. And so in RNA, it's adenine, guanine, uracil, and cytosine. So we've got cytosine, guanine, adenine, and uracil. If you look at DNA, the bases that we have are cytosine, guanine, adenine, and thymine. And so this is another difference between the two. In RNA, it's going to be a uracil. In DNA, it's going to be thymine. But if you look at the structure of those two chemicals, it's almost identical. And so they serve the same purpose. DNA is simply a double helix, so it's going to have bases on either side. And so if we have an A on this side, then we'll have a T on the other side.
The next subject that we're going to focus on is reproduction. So here I have a picture showing me some cells that are going through the process of mitosis. So what I want you to do is to draw in what's going on inside of the cells and label with what the different phases are. So here I've got the first part of mitosis. So the first part of mitosis is going to be prophase. So in prophase, a couple of things happen. The DNA is copied, condensed, and now we can see it visibly in the cell with a light microscope as chromosomes. Next part of the phase is going to be metaphase. So the key thing about metaphase is that all of my chromosomes are going to line up in the middle. So there's my first chromosome, there's my second chromosome, and there's my third chromosome. So at this point they've all lined up in the middle. And the way that those have lined up is because these little fibers have attached to each of the chromosomes that have kind of pulled them back and forth until they are in the right position. So that's metaphase. Next phase is going to be anaphase. So in anaphase, that's where those chromosomes break off into half, and then they start moving towards opposite ends of the cell. So there's my chromosomes. They've been broken in half. Again, those spindle fibers are responsible for pulling these chromosomes to the opposite ends of the cell. And the last phase is telophase. So in telophase, that's where my chromosomes have now separated to the different halves of the cell. By the way, this is a complete set of chromosomes, so it's all of the genetic information that this cell needs, these two cells need. We're also going to see the reformation of that nuclear membrane, which we see during prophase. So this is going to start to kind of form this membrane around here, and this other membrane is also starting to form. And we're starting to see those cells starting to cleave in half. So the key thing to remember about prophase is that that's where the cell prepares for division. In metaphase, we meet in the middle. In anaphase, we move away. So those chromosomes start to move away from each other. And in telophase, that's where we start to see that there are two cells. The next question asks us to compare and contrast mitosis and meiosis. So these are both strategies that a cell uses to replicate one form or another. The function/purpose of mitosis is to produce identical cells. Whereas in meiosis, our purpose is a little bit different. In meiosis, we are making unique gametes. So the purpose is to make unique gametes. In mitosis, the chromosome number is going to be what we call 2n. So if n is number of chromosomes, this would be 2. So in humans, each of our cells has 46 chromosomes. So if our cells undergo mitosis, all of the cells that are produced are going to have 46 chromosomes. In meiosis, it's actually a haploid number, n. So if the diploid number was 46, then the haploid number is going to be 23 in humans. So in mitosis, all of our somatic cells would have 46 chromosomes, whereas our gametes would have 23. Whenever we compare the genetic makeup to the parent cell, we're going to find that for mitosis, they're going to be identical, which means same number and type of chromosomes. So all of these chromosomes are going to be identical to whatever the parent cell was. In meiosis, the goal again is to make gametes with unique cell DNA combinations. So these are going to be not identical. They're going to be unique new combinations. of the parent cell. The type of reproduction used in mitosis is going to be asexual. So this would be, for example, budding, which hydras undergo. And in meiosis, we're going to have sexual reproduction. So that would be like making sperm and eggs. Next question asks us, what are the benefits and drawbacks of sexual and asexual reproduction? So, benefits of asexual reproduction. Well, for one, it's going to be fast. Bacteria use binary fission to reproduce, and in only a couple of hours, a colony of bacteria can vastly increase its numbers. So you can go from a couple of bacteria to millions of bacteria. It's very fast, and generally speaking, it's easy. It doesn't require very much energy. So the parent organism doesn't really have to invest much energy. So there's a low investment by parent. Some of the drawbacks here are that all of those cells or whatever organism, they're all going to be identical. So this is bad if you have a bunch of organisms that, say, live in one area and some kind of blight or sickness affects that area. All of those organisms are going to have the same weaknesses. So if they are weak to that disease, then the entire colony or the entire population may die off at once, and that organism might cease to exist in that particular area. For sexual reproductions, the benefits are that we're going to get that genetic diversity. So the organisms produced from sexual reproduction are going to have new genes that are going to be different from the parent. In this case, the parents, because we need two parents in sexual reproduction. So things that do sexual reproduction are people, flowers, whenever they produce pollen. Those are all sexual reproduction. Asexual would be like a bacteria or a hydra. As far as drawbacks of sexual reproduction, it requires a lot of energy. So these organisms which produce sexually, it takes them a lot of energy to produce gametes, to attract mates, to raise offspring. All of this requires a lot of investment by the parent. And if something happens to your offspring, then that investment has been lost. So the next picture asks us to draw and color code a chromosome that has undergone crossing over, and to ask us what the purpose of crossing over is. So let's say that I've got two chromosomes here. I'm going to color code this chromosome as yellow. So this chromosome is my yellow chromosome. So I have two different copies of a chromosome, and the same genes are found on each copy. So in my cell I have a chromosome that's for hair color. There'd be one for dark hair and one for blonde hair or something like that. I have two alleles for each chromosome. So I'm going to color code my other chromosome here red. So again, these chromosomes are identical. They have the same genetic information stored on them. They have the same DNA. They just have different alleles. So these chromosomes are identical. So there they are color-coded. During crossing over, that's where during meiosis these chromosomes get really close together and they can actually trade DNA. So we're going to have some sections of DNA that are going to get swapped where these chromosomes are overlapping. So here I've got my red chromosome and it's overlapping my yellow chromosome right around here. There we go, get some yellow. So here's where these two chromosomes are going to cross over and swap parts of their DNA. So the end result of this is I'm going to have two chromosomes which have traded off portions of their DNA. So my chromosome here, my yellow chromosome, is going to look pretty much the same, except for at these two parts right here, I'm going to have some of the DNA from my red chromosome. So these parts right here are going to come from my red chromosome. And on my red chromosome, I'm going to have sort of the opposite, where the parts that it lost and it traded with the yellow chromosome are now going to be swapped with yellow. So there we have it. These two chromosomes have swapped parts of their DNA. So the main thing to look at here is what we started with, two chromosomes, and what we end with is two chromosomes. And they look different than how they started. So the other purpose of crossing over is just another way that we can ensure more diversity in gametes. So even if I produce many gametes, they're not going to be all identical to the cells that they were made from. So there's this chance through crossing over that I get even more diverse gametes. And there they are, my more diverse gametes. Next subject that we talked about was genetics. So this was, remember, we were talking about genetic engineering and Punnett squares and pedigrees. So first I asked you to give me an example of genetic engineering and to explain what it is. Genetic engineering is nothing more than moving around genes. It doesn't necessarily mean that it's for our benefit or for our harm. It just simply means that we're moving around these genes. This could have huge consequences or no consequences. All that genetic engineering is just rearranging those genes. An example could be rice, which has been genetically engineered to produce vitamin A, which is an important vitamin for vision. So this could be an example where genetic engineering produced a positive outcome. We've made rice that has a vitamin in it, so people who are lacking in this vitamin, such as people in malnourished countries, they can eat this rice and it can help give them the nutrition that they need. And there's a couple different varieties of rice, and those are all natural varieties of rice. But I could genetically engineer one of these natural varieties to be a different color, or produce a vitamin, or have fewer or more calories, that sort of thing. These are all possibilities. Next question asks us to make a Punnett square showing a cross between two heterozygous pea plants. So in pea plants, round seeds are dominant to wrinkled seeds. And I want to know what fraction of the seeds are going to have smooth seeds. So first thing I got to do is I got to pick a letter. So I'm going to pick R. And capital R is going to be for the dominant round seed. And lowercase r is going to be for the wrinkled seed. So the question asks me to show the cross between two heterozygous plants. So heterozygous means different. So in this case, they're going to be different alleles. So they're going to have a big R and a little r. As compared to homozygous, which would mean two of the same. So that would be like big R, or little r, little r. So in my heterozygous cross here, I'm going to go ahead and fill in my squares. So I've got big R, big R. This corresponds to a round seed plant. Then I'm going to have big R, little r. This plant is still going to have round seeds because round is dominant. So if I have a capital R, it doesn't matter what the other letter is, it's still going to be round. Same thing here, we're going to have another round seeded plant. Now this last plant here is going to inherit two of the recessive genotypes, or excuse me, the recessive alleles. So that's little r, little r. So this plant is going to be our wrinkled seed plant. So the question asks, what fraction of the plants have smooth seeds? Well, that would be 3 out of 4, which is 75%. Next question again asks us about pea plants, but this time we're talking about the tallness of the plant. So the tall plants are dominant to short plants. So I want to see a Punnett square between a homozygous tall plant and a homozygous short plant. What fraction of the plants are going to be carriers of the short gene? So once again, I need to pick a letter. So in this case, I'm going to do T. So T is going to be tall, so capital T is the dominant trait, so that's tall. Little t is going to be short. So that would be the recessive allele. So I have a homozygous short plant and a homozygous tall plant. So a homozygous tall plant, that's going to be two of the same, and since it's tall, it's got to be big T, big T. Homozygous short plant, that's two of the same, but in this case it's got to be little t, little t. So let's do our cross. So I get a big T, little t, once again, and a fourth time. So all of these plants are going to have the tall phenotype. Big T, little t is a genotype. Tall is a phenotype. So it asks what plants are carriers of the short gene. So all of my plants have this big T, little t, which means that they carry the short gene. So 100%, or 4 out of 4, are going to be carriers. So that means if I were to take any of these two plants from this first generation here and cross them, I would see that short gene reappear in the phenotypes. Next question is about hemophilia, which is a recessive sex-linked trait due to a chromosome mutation on the X chromosome. So, I want to see the results of a mating between a normal, non-carrier female and a male with hemophilia. So once again, we need to pick a letter. So I'm going to have capital H as normal, and little h is going to be the recessive hemophilia. So first thing we got to do, we've got a man. So a man is going to be XY. This is sex linked, so I got to make sure that I write the sex chromosomes. And a female who is XX. So we have a normal non-carrier female. So big H, little h, that would be carrier. So normal non-carrier would be big H, big H. We also have a male who does have hemophilia. So for him to have hemophilia, that means his X chromosome must have a lowercase h. So now let's do our cross. So first we get a girl who is a carrier, another girl who is a carrier, a male who does not have hemophilia, and a male who does not have hemophilia. Males cannot be carriers of the disorder because they only have one X chromosome. They have it or they don't. So in this case, from this Punnett square, I'll see that none of their children have hemophilia. So there's no children who have the disease, but all of the women, all of the daughters, all women are carriers. So if one of these women were to marry a man who does have hemophilia, then their children would be highly likely of having hemophilia. Next question shows us a pedigree for sickle cell anemia in a family. I want you to use big A to represent the dominant allele for normal blood and little a to represent the allele for sickle cell blood. So then I want you to fill in the genotypes for as many as you can. So in a pedigree, remember squares are men, circles are women. If the square or circle is shaded in, then that means that the person definitely has the disorder. Now in this case, since it is recessive, I know that everybody who's shaded in has to be little a, little a. So now my job is to kind of fill in the rest, which aren't so obvious. Let's go ahead and try to figure out this parent over here, this female. So if she was big A, so she could either be big A, or she could be big A, little a. If she was big A, it would be impossible for this child right here to have hemophilia. So she can't be big A. The only option she can be is big A, little a. So if I do a cross here, this shows how some of her children could have hemophilia, or excuse me, how some of her children could have sickle cell anemia, whereas the others do not. So that means that all of these other children here who do not have sickle cell anemia are all going to be carriers, because that's the only combination possible where they don't have the disease from these two parents. So now I got to go and I got to figure out the older generation here. Now there's a couple things we got to figure out. First, we got to figure out what combination of these two parents would allow them to have some children who do not have sickle cell and some children who do. We also know that none of them are little a, little a. So we have options. We could have big A, or we could have big A, little a. If one or both of the parents is big A, I could not have this child here who has sickle cell anemia. So that means this combination isn't possible. So the only combination that's possible is big A, little a. So in that case, you would see that most of their children, 3 out of 4, would not have sickle cell, but there is a possibility 1 in 4 that one child would. So for this particular individual here, I don't know if they are big A, or if they're big A, little a, because both are possible combinations from the cross that we did between the parents. So they could either have this genotype or this genotype, but I can't really tell which. Now, if this particular individual had children, I could look at the genotypes of the children to figure out whether or not this individual was big A or big A, little a. But until I have more information, I can't be sure. The next topic that we're going to discuss is experimental design. So first I want you to define a controlled experiment and tell me what the purpose of this control is. So a controlled experiment, I need to make sure that the only thing affecting my independent variable, that my dependent variable is my independent variable. So let's say that I was conducting an experiment where I was trying to measure the effect of different colored light on the growth rate of plants. I must have a control. It's something that I'm going to compare my data to. It's the normal response or the normal conditions. So with my plant experiment, I may have made the hypothesis that green light will slow the growth rate of the plant. So whenever I take my data and I measure my growth rate over time and I look at my different colors of light, how will I know that green light caused it to slow down? I really have no way. So what I need to do is have something to compare that to. So that would be normal light. If I look at how my plants grow in sunlight and I compare it to how it grows under green light, I might be able to say, oh, well, in green light it grew slower, and I could prove my hypothesis that way. It is not what you can quote-unquote control during the experiment. A lot of people make this mistake. The control is what we compare our results to. It's how we know whether or not green light caused slower growth rate or faster growth rate, or it had no effect. It's what we compare our results to. It's not what we can change or control during the experiment. Next question, why is it important to have many trials during an experiment? So many trials is going to reduce the possibility of bias. It's also going to give me more information to base a conclusion off of. And it will also test our procedure to ensure that it has been properly developed. So the more trials I take, the less likely it is that I have something weird going on that's going to skew my data and then cause me to make the wrong conclusion. Next question, how many variables should you test when designing an experiment? So you should only test one variable at a time. Otherwise, how do I know that the dependent variable changed only when the independent variable was changed? So there should be one independent variable and one dependent variable. So in my plant experiment, my independent variable is the color of light, and my dependent variable is the growth rate of the plant. So I want to make sure that my growth rate, my dependent variable, that only changed because my independent variable, the color of light. So everything else must be constant. So I want to have the same plant from trial to trial. I want to have the same amount of water, the same soil, the same pot that the plant is grown in. All of these things need to be constant so that whenever I collect my data, I can in fact say, yes, the green light caused the plant growth rate to slow down, or it had no effect, or whatever. Just so you guys know, your benchmark will be next class. If you are absent, you need to plan on staying after school on Wednesday, Thursday, or Friday in order to make that up. Otherwise, it's going to have to go into the gradebook as a 0. The market period closes on Friday, so I need to have all of those grades in.